Look at this leaf. Over time, it dries out, curls up, and eventually disintegrates completely into dust. For most of the 20th century, scientists assumed our bodies decayed in the exact same way. The consensus was that aging was a hardware problem, driven by irreversible mutations and permanent damage to our DNA. Because of this assumption, modern medicine largely gave up on treating aging itself. Instead, doctors and researchers focused entirely on managing the downstream symptoms, fighting isolated diseases and cellular failures one by one as they appeared. If aging is truly a deletion of our foundational genetic blueprint, then finding a cure is mathematically and biologically impossible. You cannot rebuild a house if the blueprints are burned. But then a scientific discovery challenged that mutation theory: the successful cloning of an adult animal. Take a cell from an old, dying animal and use its DNA to grow a clone. The new animal is born young, healthy, and pristine. This creates a specific logical deduction. It means the digital genetic code inside our oldest, most degraded cells is actually perfectly intact and uncorrupted. The original information is all still there. This leaves us with a mystery driving modern biology. If our digital DNA hardware isn't broken or mutated, what is actually causing our bodies to fail as we get older? To answer that, we have to zoom past the DNA double helix and look at the epigenome, the body's true command center. Our DNA is made of digital information, a static sequence of letters, A, C, T, and G. But the epigenome is analog. It consists of physical proteins that spool that DNA thread into three-dimensional shapes. This mechanism physically hides or exposes specific genes to dictate what a cell becomes. The thread tightens to obscure certain traits and loosens to activate others. Think of the genome as a grand piano with a fixed set of keys. The epigenome is the pianist sitting at the bench, deciding exactly which song to play. But there is a flaw in this system. While digital data can be copied perfectly forever, analog systems are highly vulnerable to physical degradation and accumulated noise. The guardians of our epigenome are a family of ancient survival proteins called sirtuins. Sirtuins are enzymes that control gene expression and protect our cells from stress. During peacetime, sirtuins sit on specific genes, holding the DNA spools tightly closed to maintain strict cell identity. But when DNA physically breaks from cellular stress, the sirtuins immediately drop everything and rush to repair the break. This creates a long-term glitch in the system. After millions of microscopic emergencies, sirtuins get lost. They fail to return to their original posts, leaving the original DNA loop permanently unwound, messy, and exposed. Returning to our musical analogy, it's as if the distracted pianist starts hitting the wrong notes. Without the sirtuins holding the sheet music together, cells forget their assigned identities. The singular underlying disease we call aging is this specific, systemic accumulation of epigenetic noise. The biological blueprint survives the aging process. The real issue is the accumulation of analog noise that makes the genetic code unreadable. To understand this, picture a heavily scratched DVD. The reality of that DVD is that underneath the chaotic surface damage, the original, pristine digital movie is still perfectly preserved. It is just waiting to be read. The medical approach shifts when we realize our youthful data stays locked inside ourselves. We don't need to rewrite human genetics. We just need to bypass the analog noise. Scientists are currently using targeted biological stressors, like fasting, and novel molecules to wake sirtuins up and force them home. This biological process acts as an epigenetic reset. It forces the body to remember its youthful state and functionally reverse the symptoms of decay. By treating aging as a reversible engineering flaw rather than an inevitable biological law, the timeline of human longevity shifts completely. The first generation of humans to live vibrantly past 120 may already be alive today.